So, good morning, all the participants, the students, faculty members, and the invited guests. So, thank you for joining in this August occasion of a webinar, which we are organizing in a serious way. So, today the topic is, and we have started with the research-oriented topic, or that is science-oriented topic. Earlier, we had career or pharmacy practice related topic. So the aim of this topic is many of the B pharmacy students who are interested in the research. So by attending such a seminars, so it may inspire the students. So it, all the participants, I request this as one of the inspiring lecture. By attending this type of research-oriented topics, the students who wish to continue their career in the MPharm, PhD, and those who are already in post-graduation who wish to continue their research work, it may be one of the inspiring lecture. So with this mindset, we invited Dr. Murali. He has readily accepted our invitation without any hesitation. So thank you, Dr. Murali, for accepting our invitation and sparing your valuable time. On this juncture, I thank our management, Sri MN Raj Garu, and our Vice Chairman, Sri Ravi Verma Garu. They always very big boost to do all sort of these activities which will enrich the knowledge of the students and faculty members. So I thank all the faculty, student members, and all the participants, and I welcome you all to this gathering. Now I request Dr. Saranan, Professor of Pharmaceutical Chemistry, to introduce the speaker and say a few words about the today's second. Dr. Saravanan, sir, you can continue.
Yes, sir. Thank you, sir. On behalf of the MNR College of Pharmacy, I wholeheartedly welcome that all the delegates for the international webinar on role of the brain's garbage man in the nose to brain transport of polymeric nanoparticles by a well-known resource person, Dr. K. Murali. And I'm very happy to introduce our college in front of these gatherings. So MNR College of Pharmacy is established in the year 2004. It is approved by Pharmacy Council of India and AICTE. MNR College of Pharmacy is affiliated to Osmania University, Hyderabad, and we are offering the courses like B Pharmacy, PharmD, MPharm, and PharmD PB. And we have a state-of-art pharmaceutical laboratories, and we have libraries with more than 10,000 volumes of specialized books, and we have subscription to many national and international journals, and we have a digital library facilities also for our students as well as faculties. More than 50 faculty members are working in our MNR College of Pharmacy. Among them, 20 faculties are with PhD. And the faculty members as well as the student teams are led by the well-known pharmaceutical chemistry member, that is Dr. V. Alagarsamy, who is the convener and principal of MNR College of Pharmacy. And our principal is a recipient of Young Scientist Award from the DS3 India. And he published many reputed books, and the most important one is a textbook of medicinal chemistry by Dr. V. Alagarsamy. And he is the editor of Anti-Infective Agents and a well-known Bentham Science Journal. And along with that curricular activities, we are encouraging our students for participating in co-curricular activities such as attending conferences, guest lectures, and industrial visits. And we are also encouraging students to participate in the extracurricular activities such as cultural and sports activities. And with the leadership of our chairman, Sir Sri M.N. Raju Garu, and with the support of our vice chairman, Sri Ravi Verma Garu, that we are taking on a special initiatives in our MNR College of Pharmacy for university trained coaching, and we have placement facilities, and we are providing the students for a personality development, and we are giving a career counseling for our students, and we are coaching for a higher studies such as NAPER, GRAD, GPAT, TOEFL, GRE, and IELTS. And we are guiding our students for foreign university admissions also. And always our chairman tell that education and education is the two eyes. So in that basis, we are providing a favorable environment for studies in our MNR College of Pharmacy, and in our campus there is no distraction for studies, and individual care is taken for each and every students, and we are following SMS system for absentees, and every month a progress reports are also sent to the parents, and we are encouraging the students self-respect, and we are offering a education with the less cost, and we are giving a fee concession for the meritorious students also. So with this small introduction about our college, now I am happy to introduce the today's speaker to the audience. So the today's speaker is that Dr. Murali Kumarasamy. He is a pharmacist and he explore different disciplines of nanobiotechnology and neural cell biology. He combined these transgalactic ideas to initiate path in high reward entrepreneurships. So Dr. K. Murali completed his B Pharmacy in the year 2005 in the Tamil Nadu Dr. MGR Medical University, Chennai. In the year 2008, he completed his PG degree in pharmaceutical chemistry background in the same university, Tamil Nadu Dr. MGR Medical University, Chennai. And he awarded his PhD in theoretical medicine in the year 2015 in the Doctoral School of Semmelweis University, Hungary. And he undergone a postdoctoral training in animal biotechnology career enhancement program in Tamil Nadu University of Veterinary and Animal Sciences, India. And he got a postdoctoral fellowship in the Center of Nanotechnology, Indian Institute of Technology, Roorkee, India. And if you are looking at his experience, so he started his career as a lecturer in the year 2008, and around 2 years he worked as a lecturer in CL Baidmetha College of Pharmacy, Chennai. Later he got on a research fellowship in the research group of Professor Ciaro Sullivan of Nanobiotechnology and Bioanalysis Group in the University of Rovira Belgrade, Tarragona, Spain. And he done his research fellow in the topic of optimization of separation and isolation of circulating cell-free fetal DNA for non-invasive prenatal diagnosis. On basis of this research fellow, he got an opportunities to work in the Hungarian Academy of Science, where he joined the well-known European Commission FP7 Merit Consortium for nanotasks, and he completed his doctoral thesis, which he investigated on interdisciplines with neural stem and tissue type cells. And he's an early stage researcher in the Mary Curie Fellowship. So in this fellowship, he visited and made short-time research at many universities and research institutes. And he visited Norway, Germany, Spain, Ireland, and Belgium on this Mary Curie early stage researcher fellowship. And after completion of his PhD, then short term he worked in Budapest University of Technology and Economics under the professor of Dr. Istvan Esmarki, and he worked on that magneto-optical diagnosis of malaria. And Dr. Murali has around 13 years of research experience in his field, and his areas of research interest the different novel nanodrug delivery system to target different biological barriers, including blood-brain barriers, blood-fluid barriers. He is holding a very prestigious fellowships such as Mayfield Fellowships and National Institute of Health Fellowships. He published more than 30 well-known international journal publications, and he is also having a few patent applications in his field also. So from 2017 onwards, currently he is working in the Planning and Budget Council Fellowship by Israel Council for Higher Education. at present that Murali is working as a senior researcher in the Israel as well as in the International Lipid Nanotechnology Laboratory Portugal. And his current research is creating personalized solutions that utilize micro and nanoscale technologies to enable a range of theranostics for neurodegenerative disease and
Dr.
brain
Saravanan, sir,
cancers.
voice
So
is
with
breaking,
this,
sir.
Sir?
Voice is breaking. We'll ask Dr. Murali to start the session.
Yes, sir, completed, sir. So with this small introduction, I'm happy to welcome our speaker, Dr. Murali, to present his valuable research to the audience. Dr. Murali, now the stage is yours. Please proceed.
Long back, as I remember. And once again, I thank for this kind invitation. I also thank all the participants, and I thank MNR College of Pharmacy for this kind invitation. A few days back, Dr. Saravanan, he contacted me to give a talk. So then immediately I thought of giving a nose to groin transport for two reasons. One reason is nose to groin is like one of the booming area of research, especially to deliver the drugs to the groin. And next thing, recently the coronavirus is I think we are facing, world is facing unusual time because of coronavirus. This coronavirus also spreading through nose to groin and nose to lung, actually. So these are the two reasons I chose this topic. So I hope you're all safe in this pandemic. So today I'm going to talk about the role of the groin scavengement in the nose to groin transport of polymeric nanoparticles. Actually, I think later of my presentation you'll understand why I kept this funny title, groin scavengement. Most of the men, they ask why garbage men, why not garbage women, actually. Okay, I'll explain you in the later of my presentation. So let's talk about the snapshot of my presentation. First, we are going to talk about CNS disorders like groin disorders and biological barriers, what is present in the groin, what is the obstacle we are facing as a researcher, and what kind of motivation actually to do this kind of research. And next, I'm going to talk about the interaction of different polymeric nanoparticles with neuronal tissue type cells. And I'm also going to show some of the dancing videos of groin scavengement. And finally, nanosafety, that's very important. I mean, nanotoxicology, because nanomedicine is not only the research point of even nanotoxicology, also the research point of view. So these are the things I'm going to talk in my presentation.
Dr. Murali?
Yeah.
Sir, your voice is breaking, sir.
I think the problem from your side, because my internet is very good. Yeah, now it's okay. Can you hear me?
Telling like that.
Hello.
All our participants is also telling, sir, not only from my side, sir. So every participants, they are telling that voice are not clear. That's why, sir.
Oh, okay. So I think now it will be okay, I suppose. Hello.
Murali?
Yeah, I'll continue. Maybe later it will be okay, I suppose.
Okay.
Yeah.
Okay.
Yeah. Thank you.
Thank you.
Okay. So these are the snapshot of a presentation I'm going to talk. So first, I'm going to talk about the central nervous system disorders. So what are the groin disorders? We know there are more than 600 neurological disorders, but we know very few. For example, neurodegenerations, Alzheimer's, Parkinsonism, multiple sclerosis, amyotrophic lateral sclerosis, Huntington's diseases. These are all comes under neurodegeneration. There are some functional disorders like migraine, epilepsy, dizziness, and the vascular disorders like stroke, which is very, very devastating disease in the world as well as in India due to diabetes and high BP and hemorrhage. Structural disorders like brain and spinal cord injury due to accidental or natural issues, and also brain tumors. So these are the neurological diseases still we are facing actually in the humankind. So if you want to treat those diseases, particularly our drug molecule should cross this brain's own security system, which is called as blood-brain barrier. The sad truth is, most of the small molecules and 100% of large therapeutics cannot bypass this brain's own security system. That's the mystery still behind this. Still, we are neurocell biologists, neuroscience researchers. They are exploring this kind of research actually. So first thing, if I talk about the barriers, the solute barriers, which is called cerebrospinal fluid. Actually, I think as a Pharma people, you may explore all these terms, cerebrospinal fluid. The unique nature of cerebrospinal fluid is shortly called as CSF. Is formed at the rate of 0.5 ml per minute in your brain, which is present in two areas. Like one is like ventricles, the choroid plexus of ventricles, and also in the subarachnoid space. Actually, in two places, this CSF is present, and its total volume is 200 ml. And another unique nature of this CSF is like every 4 hours once it will replace actually. So these are the unique nature. And researchers, they use this liquid to deliver a drug. There are some approaches to deliver a drug through CSF fluid. And another important things, this liquid is useful in diagnostic purpose also, because taking this liquid, analyzing, it gives a lot of information about the neurological disease, including tumor. So as I mentioned, like different barriers, one is like a blood-fluid barrier, which is called, cerebrospinal fluid barrier. The choroid plexus, which is called epithelial cells. The brain has two different types of cells. One is like endothelial cells, which are also barrier, and epithelial cells, which is called has a lot of astrocyte, astroglia, it's called, and also microglia. It's like a very regulated probability. Don't details because still we don't know what kind of molecule is crossed in the brain. Researchers we are exploring these aspects. If we want to talk about charge based hyperphilicity, different particles or drugs. For example, the previous conventional methods like a cerebrospinal fluid and dialysis fiber, these are the methods which we used in vitro or ex vivo to analyze the materials. And recently, detection of fluorescence particle. Actually, this part I'm going to present now, mostly detection of fluorescence particle using a different microscopic system. And there is another in vitro models. It's called the transfer models. I don't know how many of you explored these things, which is an existing model actually to test nanoparticles or drugs in vitro. Because in vivo in brine, it's very difficult to understand the mechanisms. That's why actually we are exploring in vitro models. So if we want to talk about the mechanism of the materials is crossing the blood-brain barrier, there are different mechanisms behind this. The one mechanism, the different mechanism, one is like a diffusion based on the small molecules, like less than 400 kilodaltons molecules, and paracellular transport. As you can see, it crosses the blood-brain barrier in between the cells. And transport protein, actually, there are a lot of tight junction protein. Maybe you came across a couple of names: Claudin, Oclidin, Zonooclidin. These are the transport proteins. It's available tight junction proteins. And receptor mediated, I think a lot of research is going on in this particular area, receptor mediated transcytosis. Even we are also working on a couple of ideas using a peptides or just bind the receptors onto the nanoparticle surfaces, or drug conjugates, antibodies. So a lot of research is going on this particular area, and adsorptive transcytosis. Cells will absorb your materials and it crosses the barrier. And efflux transporters, this is the main reason for drug resistance actually. It's called ABC transporters. So these are the main reasons for the mechanism of drug transport into the blood-brain barrier. And there are different approaches to deliver a drug into the CNS, the central nervous system. The current approaches, there are like these are invasive approaches, which means opening the skull, or directly you're introducing your compounds, drug molecules into the brain. So, for example, intracerebral implants. It's 2 decades or 3 decades we are using this kind of methods. I mean, we open the skull of the brain and just we are placing your drug materials directly onto the brain. So this method is really invasive, and you need an expert of neurosurgeons or someone. And transient BBB, blood-brain barrier disruption using some chemicals, so oleic acid or such chemicals we are using. This kind of methods actually. And also intraventricular infusion, as I mentioned, cerebrospinal fluid, drugs directly injected into this fluid and to reach the brain. But recent days, non-invasive, even my research goal also the same actually. I'd like to work on like minimally invasive approaches, like non-invasive approaches, without disturbing the patients, without painless delivery. That's very important. That's the target of our future research. For example, non-invasive one actually. There are a lot of methods are available: chemicals, biological methods, prodrugs, drug conjugates, and colloidal drug nanocarriers actually. So these are the things I'm going to talk in my presentation today. Also international, actually, recent days, lot of research is focusing on these particular aspects. Like international means like nose to brain transport of the drugs or nanoparticles. This is based on 2 mechanisms. One is like olfactory and trigeminal nervous system, because these 2 neurons is present in our nose to brain. It connects the outside surface to internal brains actually. So you can say, I think as a pharmacist, you must know about the advantage of non-invasive, advantage of international delivery, particularly non-invasive. I would say the painless delivery actually, this method, and avoids hepatic bypass metabolism. That's the main advantage of this delivery methods. And circumvent the blood-brain barrier, because if you inject to the intravenous, it should cross all the barriers and it should cross the blood-brain barrier. But using a nose to brain, it's not even touching the blood-brain barrier part. It directly goes to the brain actually through these 2 pathways. The previous literature says more efficient for nanoparticles is 100 to 300 nanometers actually. So even in our research also, we are also using this range of nanoparticles. And as I mentioned, international delivery has emerged as a new approach to circumvent the blood-brain barrier and efficiently deliver the drugs into CNS to treat many disorders. Actually, this is the area of research. While I'm talking about advantages, I should say about disadvantage of this method. As you know, human nose is like this nasal area is much smaller actually, so you cannot deliver drugs like milligrams of drugs actually. So you can deliver very few amounts of drugs. That's why disadvantage of this delivery method. Before going to my presentation, I kept this unusual slide actually. I think I don't know how many of you know in Nobel Prize in Physiology Medicine in 2004, which was given to the olfactory system. That's a NASA system. They awarded to Professor Richard Axel and Professor Linda Bach. They explored about the olfactory system, this nose to brain system, and I think their discovery helps us to identify many unraveled themes. For example, olfaction, like why smell is important, how smell is important. It's same like eyes, actually, how you're looking at the world. So how you're smelling each chemicals and how it gives like different smells when you smells when you inhales different chemicals actually, and how the sensory system works and how it interacts with outside world actually. And brain has no connection with outside world actually. This one system is connecting outside world, brain with outside world. So this is a very unique system, olfactory system. As I mentioned, it crossing the blood-brain barrier. I mean, the nose to brain is like one of the ideal method, but distance-wise is smaller actually. But nose to brain is very small distance instead of using intravenous. But this olfactory region also is tightly packed system actually. It has almost no extracellular space. It has like epithelial cells. It's called nasal epithelium, olfactory neuroepithelium. It's a tight junctions and mucus layer actually it present. And not only that, it has like a lot of supportive cells which is present in these regions actually, along with olfactory receptor neurons. Always olfactory receptor always neurons will not be available in a single cell. It always is wrapped up with the different supportive cells like a glia cells actually in the CNS. So if I want to talk about central neurons, it's neurons. We have billions of neurons actually connections in our body, and neurons has the multiple cellular connections, and neural networks is dynamically changing. It's like one of the peculiar system and delicate system of connections. It's most sensitive system in our bodies are neurons actually. And the unique nature I'd like to share. The olfactory epithelium has an extraordinary unique characteristics of undergoing continuous turnover throughout lifetime of organism. Continuous turnover means here I'm talking about neurogenesis actually. I mean the adult neurons will die, neuronal apoptosis, and the new neurons will create all the times until your death actually. Even recently, last year, the papers have been published in Nature. They explained about even at the age of 95, the neurons are generating actually. So that's a unique nature of our brain system, and particularly the olfactory regions also all the time neural senses like neurogenesis happening. And the lifetime of olfactory neurons is 4 to 8 weeks actually. So after 4 to 8 weeks, it dies, and new neurons will generate actually at this olfactory regions. And I kept this slide also about, but to give, just to give an introduction about laser confocal scanning microscopy. Because most of my research I used confocal microscopy because 10 years back I was working in the area of drug discovery, but afterwards I switched to different areas of nanotechnology. And my current area of focus on neural cell biology. We have to use a microscopy, different microscopy. The one of the microscopy is confocal, but we are also using advanced microscopics like the super resolution storm and palm kind of microscopes actually, which is available. I think as far as I know, very few institutes in India, they have such kind of instruments. If you want to talk about confocal resolution is 250 nanometer. Less than this, you cannot see through confocal. The main difference between the conventional fluorescent microscope and confocal, as you can
Murali
see
sir,
here. Hello.
excuse me, Murali sir.
Yeah.
Sir, sorry for the interruptions.
Yeah.
So your voice are breaking, sir. So if possible, can you use a earphone, sir?
Okay. Okay. Just a moment. Yeah.
Yes.
I can use it actually. Yeah. Just a moment. I'm sorry for the interruption. Can you hear me now? Hello. Hello. Can you hear me?
Sir, voice was very low, sir.
Oh, just a moment. I'll increase the voice actually.
Sir, not audible, sir.
Yeah, now it's okay.
Now it was somewhat okay, sir.
Okay, thank you so much. I can choose. I hope it will be okay now.
So now the voice was clear, sir, but it was less audible, sir.
Okay, so I will. Yeah.
Because we are getting a comments that excellent presentation, but only thing is that voice was not clear like that many people.
I understand.
So that's why, sir.
Okay, I understand. So now I hope it will be okay.
Yes, sir.
It's in the 100% volume actually. So.
Okay, sir.
Yeah, thank you.
Okay, sir. Carry on. Sorry for the disturbance, sir.
Yeah, it's okay. So I'm sorry for this interruption. I'm sorry, audience, for this interruption. So confocal, particularly, it's the only difference between the conventional microscope and confocal. Confocal, it focuses on specific part of the samples actually. In a conventional method, conventional microscope, it spread, light will spread and it gives like a bloody images actually. So that's why it's confocal actually, you know. So the resolution is 200 to 250 nanometers, but there are other advanced microscopic techniques. Those resolutions are 2 to 5 nanometers. You can able to detect like 2 to 5 nanometer size particles or any kind of information. I also kept this very basic slide. So cellular imaging, how we are imaging actually cells, you know, cells like 10 to 15 micron size. So the lines will focus cells, and the confocal is mainly for the 2D cells types, like this kind of cells actually. It will not penetrate more than few microns. It penetrate up to certain microns, and it will analyze, it will give informations of the cells actually. So that's why we are getting all the images, fluorescence images and sort of images actually. So these are very basics about microscopes. And if I want to talk about I'm coming back to the main research part actually, because I gave a lot of, kind of introductions about nervous system and what kind of cells present in the nervous system. Now I'm going to give a some snapshot of my research actually we have done. Why is garbagement, what is a part of this, and nose to brain transfer things. So before I begin this research, I'd like to say some points. Actually, previously, I think this is the first report we produced. Isolated cells from the different particularly the olfactory tissues, and we performed with the different nanoparticles. Actually, the previous research, a lot of people are working in the past 2-3 decades after the Nobel Prize winning. In nose to brain transport, but mostly the people, they used directly animal models. Actually, using animal models, as I mentioned before, you cannot identify the mechanism of the diseases, particularly central nervous system. As you know, a brain is like a really complicated organ, and with a lot of billions of neurons and a lot of information. Still, mysteries are going on actually around brain. So in order to understand the mechanisms, the real in vitro models are needed actually.
So when we submitted the grant, actually, we thought of using just some ex vivo models actually. But after we approved the grant, we thought of exploring different cell types because based on my background, as you know, I'm a pharmacist. I worked in a pharmaceutical chemistry, like medicine chemistry, and I have also background of neural cell biology. Then I was discussing with my ex-boss from Technion, National Institute of Technology. Then why not we can explore this actually. Then I was going through the literatures, particularly the interdisciplinary literatures. I couldn't find any information about the materials and this olfactory cells combinations actually. So then we decided to explore this aspect. So we thought it's a novel, and also it gives a lot of information, just like introducing the materials directly into the animals and giving results. So we isolated OE means olfactory epithelium, olfactory bulb, and brine part, actually cortex part, different type of cells, and we use different specific antibodies, like specific biomarkers to identify the tubulin, like neurons, the macrophages, microglia, which is unique in your brain actually, then astroglia, then olfactory receptor neuron specific, and microglia. So these are the cell types we isolated and we characterized thoroughly. First, I'm going to talk about the characterization part. Like, so far I discussed about how we isolated, and now I'm going to talk about the characterization part, like how we isolated. Once, if you isolate cells, it is like the same like a chemistry. Like if you synthesized molecules, you're characterizing to know the structure of the molecules, right? In the same way, if you isolated cells, you should characterize the cells using a different techniques like immunostaining. Immunostaining means there are antibodies which is more specific, like biomarkers for analyzing a particular proteins actually. So as you can see in this picture, GFAP, which is called glial fibrillary acidic protein, which is more specific for the astroglia, and beta-3 tubulin, it's more specific for the neurons actually, and DAPI, it stains the nucleus, the blue color. So as you can see, when we isolate it, it's a mixed cultures. When we isolate it from the olfactory bulb of rats, it is a mixed culture. It has like a very less amount of neurons and a lot of amounts of glia cells actually present. Then we separated this the neuron specific cells using a defined culture media, and as you can see, there's the purified olfactory neurons, which is a positive for the beta-3 tubulin, and this more specific for the G protein olfactory receptors. It's also more specific actually, and this beautiful pictures. It talks about it explains about the neural growth factors actually. So it's positive for the neural growth factors. Not only that, we also did blotting analysis using a Western blot, and we found OMP, which is called olfactory marker protein, which present only in the matured olfactory neurons, and it's also present in tubulin oxygen. And we cadherin, I think I discussed about tight junctions, right? This is one of the cadherin is one of the tight junction proteins actually present in the brain. And EBA, it's a more specific for the microglia. Microglia, that's the microglia I mentioned, like a garbage man actually, because microglia is a macrophage, CNS macrophages. So it has like a unique features. Whenever the materials, I'll go back and show you some of the images actually. Whenever the materials targets the brain, whenever any materials targets brain, this particular microglia, it comes and eat up the particular harmful materials actually. That's why it's called macrophages. Macrophages means the eat up cells actually. So this this particular microglia, it acts as kind of supportive and is guard actually. It's immune sentinels for the brain environment actually. So that's specific for EBA specific. And we also characterize the olfactory epithelium. As you can see, this morphology is like a nicely tightly junctioned elongated cells. Is DAV indicated like the days actually, the 5 days, 9 days, and 15 days. And it also the positive for the olfactory marker proteins. As you can see, the neurons actually, it also positive for the other tubulins in these regions. So after characterizing, in the picture, like the funny rat picture I shown, brain regions, we isolated the primary cultures, brine cultures, which comprised of different cell types. Astroglia, the GFAP specific astroglia, and tubulin specific is a neuron specific tubulin, and O4 is oligodendrocytes, which is called macroglia. This is microglia and this is macroglia. Why the name came? Because of size actually. Small size cells are very smaller in size, but it does lot of works actually in the brain. But this cells are like a macroglia. All these cell types, it wrapping the neurons actually. Neurons are not alone, as I mentioned before. Neurons always wrapped with the different sort of cells actually. I will show you some couple of images later slide, how it is wrapping and kind of things. And another thing, before going to the nanotechnology, why the nanotechnology is important actually. I think we are in the past 15 years, this term we are hearing this nanotechnology, nanomedicine, and nanofabrications kind of thing. Even 10 years back, I was fascinated about nanomedicine, neuroscience. So why nanotechnology? What is place in nanotechnology, in particular in medicine field, particularly in drug delivery field? If you see, you know, as a pharmacist, you know the biopharmaceutical classification system 1 and 2, 3, 4. In this 4 category classification system, the 2 and 4 has an issue actually, the bioavailability issue. In pharmacology term, you know, we used to say like a bioavailability, because of low solubility and low permeability actually. So this low solubility, pure aqueous solubility, you know, our body has a lot of barriers, not only blood-brain barrier, even skin barrier and intestinal barriers, ocular barrier. If you want to treat eye diseases, ocular barrier, there are a lot of barriers actually in our body. So in order to treat these barriers, in order to cross the barriers, the drug should be like highly permeable. At the same time, it should be like a good solubility actually, because inside cells, you know, it should be soluble actually. That's why nanotechnology platforms for increasing drug resolution, improve bioavailability and biodistribution of the drug due to its unique size actually, because you know our body, lot of things in our body is in the nano size. For example, DNA is nano, proteins are in nano size. So nanotechnology also plays a very important role. Even these days, lot of research is going on in drug nanoionization, like pure drug nanoparticles, drug, just converting the drug itself into the nanoparticle, nano size. So and another sad truth I'd like to emphasize here, approximately 50% of approved drugs and approximately 70% of novel drugs fall in this category. So that's the reason most of the drugs cannot cross the blood-brain barrier or any sort of barriers actually. We are facing a difficulty in treating neurological diseases. And if we want to talk about nanomaterials, different nanoparticles is available actually. So I'm producing very few of them, but there are a lot of physiological drugs and a lot of things that are available in the research area. For example, liposomes. I think many of the people they're exploring the liposomes. Dendrimers, polymeric moieties, gold nanoparticle, polymeric conjugate, carbon nanomaterials. So carbon nanomaterials case, there are a lot of different types of carbon nanomaterials like graphene, like carbon nanotubes, boron nitrides, a lot of things actually. But it also controversial, a lot of controversy is going on between material scientists and biologists actually, because biologists are saying these carbon nanomaterials are not biodegradable. But at the same time, even in our research, we used for a tissue engineering, neural tissue engineering purposes, due to its unique nature actually. Liposomes, I think a couple of marketed drugs also available. Dendrimers also another controversial one. There are different generation of dendrimers available, but dendrimers are also slightly toxic due to its amine group present in the dendrimers. And polymer conjugate, polymers with a different targeting agent, imaging agent, antibodies, peptides, and etc. And polymeric micelles. But my current focus, I'm going to talk about the polymeric micelles, polymeric nanoparticles in my next few of my slides actually. So these are the polymeric nanoparticles we used for our nose to brain transport of I think already I discussed like what are the cells types we use. Now I'm saying like what are the nanoparticles we use actually. We use 5 different nanoparticles in a different size from 17 to almost 500 nanometers actually. Why we use this size? I think I mentioned, you know, nose to brain is 100 to 300 nanometers, so ideal to cross the nose barriers. That's the reason we explored the wide range of different size nanoparticles. And cross-link, I think I gave you the details. Like I didn't give a synthetic details and characterization details, but just I gave a snapshot of this nanoparticle details. For example, chitosan polymethyl methacrylate actually, and PVA, PMMA, polymethyl methacrylate. Why is cross-linked important? I would like to emphasize here. Without cross-linking, because you know these polymeric nanoparticles are not like our organic reactions. Now we are not doing any covalent bond interactions. Just we are doing a self-assembly, like you know self-assembly. Automatically it will assemble in certain temperatures, not like highest temperature, just like 60, 70 degrees temperatures. It automatically assemble. So in this case, when it goes to your body, for example, blood has a lot of proteins actually. So in the proteins, in this protein environment, these nanoparticles are not stable actually. That's why we used kind of inotropic interactions. We use sodium tripolyphosphate, it's called TPP, to cross-link so that you know the particles will be more stabilized actually. And we also use the chitosan PMMA, and HGMA is called the hydrolyzed galactomannan because it's one of the sugar polymers. In order to treat the brain diseases, you know, sugar is the main one of the main parameters actually. Also, we use a PEG, polycaprolactam. Then tetranix is one of the US FDA surfactants. Tetranic, pluronics. There are a lot of materials, different type of materials available. So, as I mentioned, you know, we used all these nanoparticles for our research. I think I mentioned the isolation of olfactory sensory neurons and primary cortical neurons. As you can see in the picture, I'd like to emphasize when we expose this different polymeric nanoparticles, I'm going to show only one picture because all the nanoparticles gave the same kind of results. So I'm going to show one pictures. This nanoparticles, I like to emphasize, these nanoparticles are fluorescent labeled with FATC or RATC, like fluorescent isocyanate or rhodamine B. Actually, this kind of, you know, the fluorescent materials. As you can see in this picture, the tubulin is more specific for the neurons, but the non-tubulin, like non-positive, I mean the positive cells, it accumulated huge amount of particles actually. Neurons did not take up any of the particles. Even in the brain cortical neurons, the same results we got it actually. As you can see, the neurons did not take up the particles. Instead of neurons, some other cell types is taken up the particles, and this is like without nanoparticles. And so we assume that, you know, in the brain there are like 5 different cell types mainly. As I mentioned, like endothelial, pericyte, astrocytes, neurons. So we assume that neurons is not responsible for the nanoparticle transport. Apart from neurons, there are other macrophages available, like glia cells present in the brain. So that might be responsible. So what we did, we isolated primary brain culture, which contains 3 different cell types actually. One is like astroglia, is one of the glia cell astrocytes or astroglia, you can say. And very small amount of neurons and small amount of microglia actually. I mentioned, you know, CNS macrophages. So this is comprised of 3 different cell types. As you can see, when you expose polymeric nanoparticles, the particular cell types, as you can see in the picture, it taken up this the particles. So the 2 reasons actually. So neurons, you know, did not take up the particles. So the other 2 things is present here is microglia and astroglia. In astroglia, I would like to emphasize here, in the mild injury or external invasion, like foreign invasion into the brain, astrocytes become phagocytes. Phagocytes means, you know, like highly eatable pharma. And it removes foreign materials to produce inflammatory, you know, it produces anti-inflammatory cytokines to protect the brain actually. And in severe cases, this astrocytes, the astroglia, will activate the microglia. So there's only two reasons, the astroglia or microglia. So then we did other experiments using a microscopy, and as you can see, the particles are internalized the cells actually. And we also performed experiments at 4 degrees centigrade. Why? Because in order to know whether it's like an active process or non-active process. For example, our physiological temperature, as you know, it's very basic. Actually, it's a 37 degree. The 37 degree, the outside temp, regardless of outside temperatures, the body, always it creates like a 37 degree. So in 37 degree, our cells are really active. All the transporters and then cell materials, cytoplasms will be active in this particular temperatures. So when you reduce the temperature, the 4 degrees, cells, most of the transporting things, active transports will be suppressed at this particular temperature. That's why we also did experiments with the 4 degree. And as you can see, in 4 degree, the particles only the outside surfaces actually, not internalized. And as I mentioned, only two cells are responsible. One is like astroglia or microglia. In this case, we also separated the astroglia from the purified cultures of astroglia, as you can see in this picture. This picture shows, one of the very few, I mean, the palpating microprocess like nanoparticles internalized in some selected astrocytes, not all astrocytes, as you can see. So we presume that so these particular astrocytes got kind of activation, but not all the astrocytes actually. We also repeated the same like low temperature analysis in order to know whether it's active or not active. So as you can see at the 4 degree, this particles are outside, it's not internalized. If you can see, in 37 degree particles internalized, in 4 degree particles are just outside actually of the cells, which means that cell is suppressed at this temperature. Cells is not allowed any of the materials. Cells are calmed down at this temperature actually. That's what I can say. So in this case, one is some selected astrocytes it taken up this the particles, not all astrocytes. Okay. And what we did afterwards, we did a separation of microglia from other cell contaminants actually, because then we realized the microglia, the garbage men of the brain, is the reason for the nanoparticle uptake. So we separated the microglia from the other cell types. I think this is a nice video I'd like to show. As you can see, using some mild trypsination, using certain media conditions, all other cell types is removed, and only the microglia stays back in this region. So this way we are separating the microglia, the CNS macrophages, or otherwise called as garbage men, from the other cell types to do some of the works actually. So as you can see, this layer of in this layer, the astrocytes neurons are present actually. So we are separating this astrocytes and neurons, and only the microglia stays back in this video. So also I like to show other dancing video of this microglia. As you can see, the microglia is a separated microglia. It eat up some kind of debris actually. You can see this is called like a phagocytosis. In other case, you can see, even this case also, you can see this microglia also, it eat up this the other debris actually. So this way, if any particles or any harmful materials if enters the brain, this microglia will chew up those materials. So that's the nature of this microglia, or otherwise called as garbage men actually. That's why I kept the names garbage men. Then after purification of microglia, as you can see, there's a microglia pictures. This a layer of things is separating only the microglia present in this regions. We again expose this polymeric nanoparticles I mentioned. As I mentioned, polymeric nanoparticles are fluorescently labeled with this green color indicates the nanoparticles, and the red color indicates the microglia, this microglia specific marker. As you can see, regardless of the cellular origin, both origin nanoparticles, the cortical and olfactory microglia, it internalize the huge amount of nanoparticles. So I think we got a conclusion at this point, like microglia is the main reason for nanoparticle transport to the brain actually. Again, I would like to emphasize here, neurons is not like a separate cells. It always wrap up with other glia cells, microglia, macroglia. So when any external materials goes without any specificity, this microglia will capture those materials. In similar way, we got results actually here. And also this particular cells is positive for the INS. INS means nitric oxide synthase. This is one of the specific biomarkers for the cells. Those are under stress actually. So this, as you can see, these cells are under stress, and it took up this huge margin. It internalized the nanoparticles. And also it's when you introduce the different sort of nanoparticles I mentioned, cross-linked, and there's all these nanoparticles. As you can see the bands, you can see some of the nanoparticles produce the positive for this INS antibodies. And further, what we did, we isolated this microglia, the specific macrophages, from the transgenic mouse, which is like, I think you like genetically modified organism mouse. This CX3CR1 is nothing but it's a fructylkine receptor. It's more specific for the macrophages. So we isolated specific microglia, the purified cultures of microglia, and we introduced the rhodamine labeled polymeric nanoparticles. As you can see, rhodamine is the red color. It's nicely decorated this microglia surfaces by these rhodamine labeled nanoparticles. So we got we concluded this microglia is the main reason for the nanoparticle transport. I mean, in my term, if I want to say, is garbage men is responsible for the transport of nanoparticles to the nose to brain. In other cases, we introduced a galactomannan is one of the sugar polymeric nanoparticles. And as you can see in the particular purified cultures, you can see a lot of dots kind of thing actually. I think before this research, I never know about this particular why dots are coming. So then I started analyzing it. Then these dots are nothing but it's a lipid droplet accumulation in glia cells, because I never exposed, even the most of the researchers never exposed the lipid droplets, lipid metabolism in a CNS brain tissue type cells. Because this lipid metabolism, it might be the main mechanism for the adipose tissues, because which has like a lot of lipids, or in some certain cases cancer cells actually, because for the defense mechanism, cancer cells will produce this kind of lipid droplets. But the first time we explored this lipid droplets, this is characteristic of lipid droplets. It contains phospholipid monolayers, lipid anchors, proteins, cholesterol sterols, and a lot of things are actually present. So when you introduce certain nanomaterials, these cells, actually the microglia cells, got kind of stress actually, which is called as lipid droplet accumulating microglia. It's called LDAM. So this stress, because of high levels of ROS, reactive oxygen species, and secrete pro-inflammatory cytokines. And further, we like to investigate using ultrastructures, using an electron microscopy. I think you may came across same scanning electron microscopy or other side of electron microscopy. As you can see in this ultrastructures, these droplets, the dot, it indicates the white circles actually. These are the lipid droplets, which is because of the cell stress. So this ultrastructures, electron microscopic images clearly indicates lipid droplets inside this microglia. And again, I would like to show this dancing videos. As I said, microglia is like a dynamic sensor. It's a sensor of the brain environment. and always is sensing the brain environment. And as you can see, this microglia is like phagocytosing different tissue debris actually, because these are the debris present in these regions. So or deadly neurons, as I mentioned, neurogenesis happening. So what will happen once neurons dies? So, after neurons death, it becomes like a debris actually, as you can see here, like this becomes debris. So these debris will be cleared by this microglia. See how it eating the debris actually. And as I mentioned, we use a transgenic mouse to isolate the microglia. These microglia is really active. That's why I mentioned dancing videos. The microglia are always in the active. It's always is monitoring the brain environment actually. So as you can see how is motile it is. So that's why I mentioned it's a never resting garbage man. It's called microglia. So and also we did another ultrastructure analysis using electron microscopy. As I mentioned, microglia are always policing. I mean, always monitoring the neuronal environment, neuronal bodies in a specialized junctions. In this ultrastructures, as you can see, this lipid droplets, which is a characteristic of this microglia and neurons, because there is a lot of neuronal bodies actually. You can see it's called Nissel bodies. So this microglia interacting with neurons at some junctional point actually. It's called pyronergic junctions. So always microglia is monitoring. If any materials is targeting this biological wires, I used to say neurons are like a biological wire because it's crossing it conducting the kind of electricity. So this biological wires, if any material is targeting this biological wires, the microglia will be activated and it protects the neurons. As I mentioned, neurons is like a really is in a delicate system and is more peculiar and more sensitive to external insults. So these are the things. And I'm coming back to the summary of my presentation. Actually, I'm going to present some of the things, but for the summary of this particular results, the inherent defense mechanisms in the olfactory regions, particularly the mild injury, astrocytes become phagocytes, remove foreign materials and produce anti-inflammatory cytokines. In case of your materials is not toxic, just very, very light toxic, the astrocytes will be activated. Astrocytes, astroglia will be taken care of the aspects. And in case of excess molecules, like any toxic materials enters brain, this reactive astrocytes will produce pro-inflammatory cytokines and it activates the microglia. This is this garbage man in CNS, or CNS resident macrophages, and produce a lot of inflammations and everything. That's the reason even in COVID cases, the coronavirus cases also, because once virus enters, I think people, they feel headache, because the headache, it comes because of this kind of inflammatory cytokines productions. So based on all the results, I hope you all understand what I explained to you. If you don't understand, you can ask me at the end of my presentation, or if you hesitate to ask any questions, you can contact me through email. Actually, I mentioned in the beginning of my slides. So our findings reveal that neither olfactory nor forebrain neurons internalize nanoparticles. Conversely, olfactory and cortical microglia, that is garbage men, phagocytose the nanoparticles independently of their futures actually. So neurons not, microglia yes. So this is the final conclusion I'm giving you at this moment. And on other hand, I hope you remember the mechanism of brain penetration. I mentioned receptor-mediated transport of nanoparticles or any drug materials. So when in the previous research, what I mentioned, neurons did not take up any of the polymer, any of the materials, any of the nanoparticles. But in this case, we functionalized polystyrene nanoparticles with cyclic RGD, which is one of the cell binding peptides. We functionalize polystyrene nanoparticles. So as you can see in the pictures, neurons, it nicely addition this nanoparticles. This green color is indicates the nanoparticles, and red is like specific for the tubulin. You know, as I mentioned, it's neuron specific, and blue is for the nucleus. As you can see, this functionalized, this peptides functionalized polyester nanoparticles, it nicely addition this this nanoparticles, which means that it comes in the receptor base, because there is an integrin receptor. It's specific for the cyclic arginine. It bounds on that particular point actually. So at the same time, I think I know many people. It's other some extra works I'm presenting you. Some of the awareness about those who are working in nanomedicine. When you are synthesizing your nanoparticles, any sort of nanoparticles, any nanomaterials, either polymeric liposomes or carbon materials, toxic contamination is the main problem actually, because as I mentioned, nanoparticle has a unique surface properties. It's a surface like really active, so it bonds any sort of biological materials. It's called bio corona. I used to say term protein corona. Actually, it's not about virus, but it's about corona means it's like a binding. You know, it's like adsorption. That's the reason even they kept the virus named coronavirus, because there is a spike protein. This spike protein is it bonds with any sort of surfaces actually. That's why it's called as corona. So the protein corona, because of protein corona means protein nanoparticle interactions, or bio corona means any biomolecules nanoparticle interactions. When you are synthesizing nanomaterials, this endotoxins, I hope you all know about these toxins, the pyrogens, particularly when you're producing a parental products, you used to use this kind of terms like toxins, pyrogen free, non-pyrogenic, blah blah. All these combinations of words actually you explored. So endotoxin is a big contaminant. It bounds on the surface of the polystyrene nanoparticles. In our experience, I'm sharing actually with you. So using a LAL assay, so I think as far as I know, your pharmaceutics, you used to study this about this assay, limulus amibicide lysate assay actually. This assay is the only assay available to analyze this endotoxin contaminations. As you can see, the carboxylated polystyrene and PEG. PEG we are saying is more safer, but PEG it contaminated more than carboxylated particles actually. So always you should be very careful when you're using this kind of, when you're producing nanoparticles. You should produce in the high clean room facilities. You have to use highly distilled water to produce, or distilled materials to produce to the nanoparticles, particularly in brine aspects actually, because brine has microglia. So microglia is responsible for these toxins actually. So that's the reason even for the corona patients, you know, it targets the macrophages, and because of macrophages, a lot of sepsis like cytokine storm happens. A lot of inflammatory cytokines produced, and so this sepsis is the main problem actually. So even for when you're synthesizing nanomaterials, you should be very careful about the endotoxin contaminations. That's my conclusion at this moment. So far, I discussed about nanomedicine point of view. Nano is not only for the application point of view, even nano is for the toxicology point of view, nano safety. For example, many inorganic nanoparticles. For example, silver. It's a toxic for the humankind, and particularly I'm talking about nanoparticles in the central nervous tissue, like brain tissue. As you may know, these days, in the past 5 years, there's a lot of products, marketed products available in the market, which contains nanomaterials. So there is an urgent need to assessment of biological effects and health risks, actually not only biological effects, and also as well as the health risks of nanomaterials, because of the acceleration of both manufacturing scales and intentions to use. Actually, even a lot of costumes, a lot of like, you know, materials like ointments and even fabrics. I saw like silver pant, a lot of materials actually which contains nanomaterials. So it's accidental exposure or manufacturing scales. So these nanomaterials it spreads in the in the environment. Even I also wrote very small articles recently. The coronavirus, it has like a spike protein, right? It's a glycoprotein. These proteins, coronavirus is also spreading in the air. So when the nanoparticles in the air, a lot of nanoparticles in the air, for example, carbon nanoparticles from the diesel materials and everything, carbon nanomaterials is spreading in the air. So this virus, it easily makes corona with the nanoparticles, and it spreads and it affects the humankind actually. I think we are new to this coronavirus, this particular area, but this is my hypothesis, my assumptions. So anything can happen actually. In case of brine aspects, as I mentioned, nose to brine, because nose is just like outside the environment, it connects the brine. So any sort of nanomaterials, contaminated nanomaterials, it can penetrate into through your nose to brine. It affects your brine actually. So routes of invasion, permeations, and distribution in various cellular tissue compartments, and effects on different types of neural tissue types. As still we don't know actually about this particular area of research, nanosafety. But still people are exploring, and European Commission has invested huge amounts, huge funding for this nanosafety. And so the last part, I'd like to acknowledge so many people actually, and my lab people in Technion. So it's a nice family actually. We enjoyed a lot of things actually together, not only in science, apart from science, we enjoyed a lot actually those times. I'd like to thank the European Commission for funding our projects, because they are funding huge amounts to explore this nano neuroscience aspects. And Marie Curie Fellowship, one of the prestigious fellowships for the PhDs or postdocs. I like to thank Teva Pharmaceuticals. I hope you all know about Teva Pharmaceuticals, which is based in Israel, but it's situated in more than 90 countries. We got this grant to explore this nose to brine transport of nanoparticles. And also I'd like to thank Council for Higher Education Israel. Israel is fantastic in doing research, and I'd like to thank Israel. And Technion which is called Israel Institute of Technology, but in otherwise is called as Mediterranean Institute of Technology. That's MIT of Mediterranean. A lot of Nobel Prize winners in Technion. And I'm always looking forward while you collaborate in successful work. You can stay in touch. You can contact me, especially students. I know Dr. Saravanan told me a lot of students are attending in this seminar. I don't know how many of you. I think I can see 293 participants. I don't know how many of you attending through YouTube. So you can, if you want some details, I cannot provide you positions, but I can guide you in different aspects. So feel free to contact me anytime. So thank you for your kind attention, and thank you very, very much for this invitation. I thank once again, I thank Dr. Saravanan and Professor Alagarsamy for this kind invitation, and I also thank the MNR College of Pharmacy Management for such a wonderful webinar series. I think this is an ideal time to explore, to share science through webinars. It's a nice interactions. So thank you so much, and all the participants particularly. I hope you understand what I explained to you. If you don't understand, please ask me any questions. I always wish to receive some questions. If I can able to answer, I can answer now. Otherwise, I can email you later actually about the answers. Thank you so much for this wonderful invitations, and thanks again.
Yes. Thank you, Dr. Murali sir, for your wonderful session.
Thank you.
So now the forum is open for questions. So if you have any questions, you can raise your hands and we will unmute you. You can ask your questions. So if anybody having any questions, you can raise your hand. We will unmute it. Yes, Mr. Gauri Radhakrishnan. You can ask your question. Mr. Gauri Radhakrishnan. So unmute yourself and you can ask your questions.
Hey Gauri, can you hear me?
Ah, yes.
No.
Yes, sir. You can ask your question, sir. So I think some disturbances in him. I think we can go ahead with the other fellows. Mr. Mohan Raj. Mr.
Yes, sir.
Mohan Raj. Yes,
Yes,
sir.
sir. Yes, sir. Good morning, sir. How are you, sir?
Fine, fine. You can ask your question, sir.
Hi, sir. Murali, sir.
Hey Morgan, yeah, thanks.
How are you, sir?
Yeah, I'm good. I'm good, Morgan Raj. How about you?
I'm fine, sir. I'm fine.
I hope you're all safe in this pandemic.
Yes,
Yeah.
yes, sir. Sure, sure.
Yeah.
I think you also safe in your there.
Yeah, it's now we are okay actually.
Okay, thank you so much, sir. I listened your valuable research
Thank you so much.
experience you shared with us.
Thank you.
So I have a doubt in that.
Yeah.
Actually, I am a research scholar. I did my research in snake venom.
I mean, I'm just kidding you actually. You have doubt in my experience or you have doubt in my presentation?
Sir, doubt in your present. I have some doubt in your presentation,
yeah,
sir.
just
I want to clear that.
Yeah,
okay, sir.
yeah, I can. Yes.
Sir, sir, how will neurons by prevent fatty acid toxicity?
Actually, as mentioned, neurons has a lot of actually the primary prevention is like a glia cells. Okay. Apart from glia cells, neurons also expressing a lot of transporters actually. For example, I mentioned efflux transporters, like it's called PGP actually, glycoproteins. Those efflux transporters, if any toxic materials goes into particular cell types, this efflux transporters is called ABC transporters. It efflux away. That's the reason actually even in cancer or any other diseases, drug resistance actually, because when you're introducing any materials, this efflux transporters will spit out actually, it spit out those materials, so enough materials will not reach the particular cell types actually. In the similar way, neurons also doing through their transporters actually. But apart from that, the glia cells glia cells, astroglia and microglia it's doing a brain homeostasis actually. It's monitoring and it's doing it's stabilizing the environment actually. So any toxic things will not target these neurons directly. But in extreme case, for example, in certain disease cases, actually neurons degenerations happen. But in extreme cases, neurons will be affected. That's why a lot of neurodegenerative diseases are affected. So I hope I answered your question.
Yes, sir. Correct. Sir, another questions.
Yeah.
Why to maintain a temperature in astrocytes? Is it only for specific nerve cells or any other than the astrocytes?
No, not for the astrocytes actually. Even for I mentioned not only for the astrocytes, even for any sort of cells actually, you can do this the temperature assessment actually. As I mentioned in my presentation, 37 is like a physiological temperature.
Yes, sir.
Most of our research, what we are doing, we are working on 37 degree. If you are working on cell biology, the very basic things, you're keeping in 37 degrees.
Yes.
In 37 degree, as I mentioned, all the transporters, cells has a lot of transporters like glucose transporters and efflux transporters, influx transporters, and etc. All the things will be active in this physiological temperature. That's why physiological temperatures. If temperature reduces, what will happen? Right? So it's not only specifically for the astrocytes; it's specifically for each and everything. You also can do same experiments for the cancer type of cells, and you can give a mechanism like your uptake or your interaction is based on active or passive, something like this.
Sure, sir. Okay.
Yeah. Okay.
Thank you, sir. Thank you so much.
Thank you. Thank you, Mohan.
Thank you, Sir Anand, sir.
So, any more questions from the students? You can feel free to ask me question. Any question you can ask, any basics. Because I'd like to share some of my experiences. 10, 12 years back, I was in the same situation like you people. I was really shy to ask questions, and even I never explored this about neurons. I know neurons, but I know neural stem cells, but I never know how it looks actually. So, but in the past 10 years, I explored all the things actually. So you can raise your hands. You can ask any questions, particularly students actually. You can.
Sir, what type of nanoparticles are used in the treatment of cancer?
In the treatment of cancer, as I mentioned, liposomes actually. I think doxil, those kind of things they used in a cancer treatment. And another case, there is a new term you may came across, photoablation therapy. Many researchers, they are using gold nanoparticles. It's like a localized delivery actually. So you can target. I mean, you can inject. You can localize these nanoparticles, and passing heat. That's why it's called like photo ablation or photodynamic therapy. So gold nanoparticles are using, and a lot of liposomes products, commercial products are available.
अरे भाई कमेटी के लिए कोई कमेटी लग रही, उसी समय क्यों हाथ पैर मार रहे हैं लोग? ये अलग से कमेटी क्यों नहीं बनवाती जाके वहाँ?
Hello, sir, am I audible to you?
Yes.
Hello.
Sir,
Yeah.
I would like to ask a question.
Yeah.
Sir, nowadays these gold nanoparticles are now being synthesized in combination of curcumin also.
Okay.
So
Yeah.
how for these combined nanoparticles are useful in the treatment of cancer and other diseases also?
Sorry. Could you please repeat? I got some interruptions.
Sir, actually, sir, I read so many articles. People are now synthesizing the gold nanoparticles, and they are combining these gold nanoparticles with curcumin.
Yeah.
So, I would like to ask you that this combined gold nanoparticles with curcumin, have they been effective for the treatment of cancer specifically and other diseases also?
Actually, the reason why they're combining, the two reasons actually. One is that's the reason term is called theranostics actually. Therapeutics come diagnostics. When you combine with gold nanoparticles with curcumin, curcumin, I think a lot of people are working. One thing, curcumin has a lot of issues, particularly the solubility issues actually. Okay? That's the reason we are making into nano. The reason why they are using a combinations, like particularly if I want to answer your question, like gold and curcumin, the reason term is called theranostics or theragnostics, like therapeutics come diagnostics. In this case, curcumin will take care of the therapeutic applications, And gold nanoparticle will take care of the diagnostics. Like for example, if you're injecting with the certain peptides, you conjugated curcumin, gold nanoparticles conjugated with specific antibodies or specific peptides to target certain cancers. It goes and bonds with the cancer tissues, so that you can able to detect the particular cancers just by imaging, nanoparticle imaging or some plasmodic imaging. And curcumin, the application of curcumin. Curcumin will treat the cancer more specifically. That's the reason they are combining this gold nanoparticles with curcumin or any sort of things actually.
Thank you. Thank
I
you,
hope
sir.
I hope you understand my answer,
Yes.
right? Yeah.
Yes, sir.
It's a combinations for the theranostic purpose.
Yes, sir. Yes.
Dr.
Hello, sir.
Murali, I am Alagarsamy.
Yes, sir. Please.
Some of the students during the presentation, they raised the clarifications. How do we apply for these fellowships like Marie Curie fellowships, all how to apply? So can you address them?
Yeah, sure, sir. Definitely. Yeah, I'm always, I wish to help our fellow Indians, particularly the pharma people actually. Also, I would like to share some of my experiences. I worked in so many institutions, and I never saw like any pharmacy people. They used to work with me actually, particularly from India. Okay, I saw like some pharmacy people in USA and some places actually, but for this kind of technical institutions, I never saw pharmacy people from India. That's a big lack. I don't know, information wise, or maybe our people are more narrow. You're not thinking more wide aspects. If you are really interested in research, as I mentioned in my bio sketch, right now I'm interested, like not interested even. I'm trying a couple of startup things actually based on my ideas. I had a lot of experiences. I worked with many laboratories, many big professors. So after my experiences, I worked in a many interdisciplinary. For example, as I started my life as a pharmacist, then I explored nanobiotechnology, drug discovery. Of course, during the time I was collaborating with Alagarsamy sir, drug discovery, and I explored nanobiotechnology, nanotechnology, nanofabrications, and also explored neurocell biology. So also I did a lot of other things like chip microfluidics and etc. So when you combine all these things, you get a lot of ideas actually. So a lot of ideas actually. You will not understand, or you will not get an answer from one field. If you work on another field, then you will come to know what is lacking in another field actually. So okay, I'm answering Professor Alagasamy's question. Fellowships actually, Marie Curie fellowships is one of Europe's prestigious fellowship. European Union is spending a lot of money on these fellowships. These fellowships both for the PhDs and postdocs. Postdocs, that's a different case actually. You need to write your own proposal. That is a really competitive proposal in a different aspects. That you need to choose a nice area of research, and that's a different actually postdocs. In case of PhDs in Europe, there is a website called Euraxis that advertise a lot of job opportunities in all over Europe, and also you can explore the same Marie Curie actions. You just type in Google like Marie Curie actions, you will find the options called jobs. You can apply any sort of jobs for a PhD's, master internships or postdocs, industrial openings, because European Union has a regulation to advertise any openings in these websites like Marie Curie, and there is another one is called Euraxis, EURAXESS, Euraxis. Apart from that, there are other websites like Nature Jobs. So these are the websites available to search for the jobs actually. In case of masters, for example, after undergraduate, you'd like to explore masters, right? There is the European Union fellowship called Erasmus Mundus Masters. List of master's courses are there actually. When I was doing my, those times, there are not many master courses actually. But these days, a lot of master courses they introduced, and salary-wise is really huge salary. Even for the masters, you get like 1,000 euro. I think that's more than enough as a single man. And is of PhDs, salary also very good. It depends on the country you get salary. For example, Germany. In Germany, there is another website called DAD, DAAD, DAD actually. You can also visit Goethe Institute actually. Every big cities, I think in Hyderabad they have a Max Muller Bhavan actually, the German Cultural Exchange Institute. If you want to do your procedural degree in like high studies in Germany, you can go to the institute. There is a seminars, you can attend the seminars. They'll give a lot of information. You get a lot of information. So what are the websites I mentioned, like Euraxis, Nature Jobs, DAD, and also you can explore each university websites. At the same time, you also can talk to professors actually, those who are coming to India. But the big problem is funding actually. We are struggling for funding, and highly competitive. We are like 28, 30 countries we are competing for a funding. So all the time in Europe, another thing is there is no permanent jobs actually, always is tenure track, because you need to prove your efficiency every 3 years or 5 years once. So you need to apply. It's highly competitive. So always we wish the candidates to get their own funding, or to apply for this kind of fundings like Marie Curie, or something else actually. So also for masters, Erasmus Mundus. In case of PDF, I think postdoctoral is not like a degree; it's kind of job actually. You also can think about industrial postdocs. If you want to explore industry after PhD, you can choose industrial postdoc. A lot of industries like AstraZeneca, Novartis, they are offering industrial postdocs. You can also choose those things. Industrial PhDs also, and I would say NUS, Singapore, NUS and NTU is Asia's top universities, top 50 universities. You also can explore in those. NUS is offering a lot of star fellowships and etc. You can explore those things. In Europe and US also a lot of, but the US problem is they don't have much fellowships. You need to go with your own funding, then they offer you. But US has a lot of postdoctoral fellowship. If your profile is very good, they offer you postdoctoral fellowship. Those postdoctoral fellowships are really highly competitive. For example, NIH fellowships, you need to submit many proposals to get such fellowships. So I hope, sir, I answered your question. Like these are the scholarships and fellowships available to explore it.
Yes, sir. Yes, sir. It's clear, sir. So, still from the participants, any more clarifications like regarding the subject or regarding this type of admissions or fellowships? Still, you can feel free to contact Dr. Murali is available for us.
Sir.
Hello. Sir.
Yeah.
Murali, sir.
One by one we will call you, please. If all you people are talking at a time, it is very difficult. So we will call one by one. Please wait. So Dr. Murali, so there is one question from a YouTube contestant. So he is repeatedly asking that please ask my questions. So the question is from Dr. Swati Mehra. So she is asking, sir, can you specify any course elaborated one to learn nanoparticle characterization and how to process out or extract informations of these sums? So she's asking about the course related to nanoparticles. Is there any course is there, and what is the process?
Actually, you mentioned she's a doctor, like she's based.
Yes, yes, she's
She-
Dr.
yeah.
Swati Mehra.
Hi, Dr. Swati. Thank you for your question. I think in this Corona is pandemic, but it helps us a lot of things actually. I mean, a lot of courses. For instance, as far as I know, the lot of European institutes, they are conducting online courses, especially on nano, not only in nano, even many subjects. University of 20 actually, they are conducting one online course actually. The course is about very basics. Familiar in nanotechnology, they are well versed in nanotechnology. They have excellent facilities. They are conducting such a courses actually, online courses. You can register in that courses. I don't know the exact name, but you can explore those things actually. They are conducting in each and every aspect. So I just glanced it actually. I don't know the much details, but they're covering each and everything from the nanofabrication point of nanomedicine, nanoparticle characterizations, and etc. I think you need to explore the internet. And other case, there is a public platform also available, NanoWork actually, I suppose. So you also can go explore and get to know about this courses available. But as far as I know, University of 20 Netherlands, they are organizing such online courses, so you can attend such courses actually. I'm sorry, I cannot answer you more, but this is the only thing I know actually at the moment.
Thank you, sir. So now that I can unmute that Manisa.
Yeah.
Manisa, you can unmute yourself and you can talk. Manisa?
Yes, sir.
Yes. You can ask your question, madam, now.
Hi, Manisha. Yeah.
तो मेरा क्वेश्चन ये था कि कैंसर के ट्रीटमेंट में कौन से नैनो पार्टिकल्स यूज होते हैं, और जैसे डायबिटिक डिजीज होगी, ये सारी डिजीज में नैनो पार्टिकल्स.
You mean for the cancer and diabetes?
हाँ sir. Yes sir.
I think for the cancer I already mentioned, but for diabetes, nano it's still in infancy actually, because there are a lot of regulatory issues in nano. I mentioned right in my last slide nano safety. Nanoparticle is not just like nanoparticles actually. Nanoparticle has a lot of issues actually. For example, aggregations, agglomerations, and there is no uniformity lab to lab. For example, if I'm synthesizing nanoparticle, if you are synthesizing the same nanoparticle, there are a lot of deviations actually. So there's a lot of regulatory issues in nanoparticles actually. That's why still nanomedicine, it's in its infancy. Very few products are available against cancer. As I mentioned, liposomal formulations. But for the diabetes, there's no nanoparticles, but the nanotechnology plays a very important role in diabetes, like diabetes diagnostics actually. Nanotechnology is not only for the therapeutic, diagnostics also important actually. So nanotechnology in diabetes, much used in diabetes diagnostics actually, not in therapeutics. And now people are exploring, but I don't know how far it's nano can cure this kind of diabetes actually, type 1, type 2, and etc. But cancer, there are solutions. There are a lot of methods, a lot of things they are exploring. As I mentioned, non-invasive is another area of research. There are few marketed available products, a lot of products in clinical trials. So yeah, Monisha, that's what I can say at the moment.
Thank you, sir, and nice presentation, sir.
Thank you so much. Thank you.
Thank you.
Hello.
Hello, sir. Sir, one more question.
Yeah, yes, Sakiran. Your name is Sakiran?
Sir, I'm Google from JK International College of Pharmacy. Sir, herbal products also go in with nanotechnology or same dosage form, sir.
Yeah, actually, herbal formulations is another way of exploring nanotechnology. For example, I know we are exploring plant extracts or Ayurvedic formulations and extracts, etc. As I mentioned, we are what we are doing. We are the polymeric mices or polymeric nanoparticles. You are just encapsulating the herbal formulations. You are making a polymer herbal formulations actually. That gives a lot of advantage over conventional herbal formulations. For example, control release. You also can go for the target base actually. I think a lot of exploration is going on, but again, it has like a regulatory issues. Not like much marketed products actually, but a lot of people are working on it. That's why even I also suggest to use people. Recently, one of my friend, he contacted me asking for applying Ayush product. Then I suggested him to use polymers to encapsulate to deliver actually, because just doing herbal formulation is it's not much useful actually. You can make a polymer herbal formulations, and you can deliver to any sort of roots actually. So that's a very good area of research actually. So local. I hope.
Sir, that product have also clinical trial.
Yeah, there are actually. There are some products are in phase 1 clinical trials actually. For I don't know exactly what kind of disease actually, but this natural products is in clinical trials actually. Certain products are in clinical trials, but it's in just initial clinical trials actually, not like advanced clinical trials.
Thank you, sir.
Another problem is,
Hello, sir.
when you are going for a clinical trials and other regulatory issues, they ask for the active constituent actually. In herbal, another problem is the whole herbal is more active because of agonist issue, but when you isolate a particular active constituent, sometimes it's inactive actually. Sometimes it's not soluble. For example, okay, curcumin, we are so lucky to have a curcumin. It has like a wide application, but you cannot expect same active constituents from all herbal things. So there are some regulatory issues also.
Hello, sir. Can I have a question, sir?
Yeah, please.
Sir, I'm working on adrenaline, sir. Its effects.
Sorry. What's your name? I'm sorry.
My name is Sai Kiran, sir.
Okay.
I'm adrenaline and its effects on the neurological effects. Neuro problem. If we have adrenaline artificially in our body, is there any effects in our brain, sir?
I cannot hear a question properly.
Hello.
Yeah. Hello.
Sir.
Hello.
Good morning, sir.
Hello.
Yeah.
This is Dr. Somati.
Hello.
Yeah. I can hear you.
Hello, sir.
Yeah.
Can you hear me, sir?
Yeah, I can hear you.
You can ask your question, madam.
Now, someone they asked me about neurons. I didn't get his question. Maybe if he can repeat his question, I can answer actually.
Sir, hello? I have one question, sir. Hello.
Yeah.
Hello.
Sir, can I complete my question, sir?
Yeah.
Hello.
Yeah. I can hear you.
So please, one by one, you can ask. If you are at a time, if you are asking, means we can't able to answer for anybody's questions. So please wait. Based on that, you are raising your hands. We are calling one by one. That time you can ask.
I'm waiting for the question who asked about neurons. Actually, I'm
Yes,
still
sir.
waiting. Yeah.
Sir, I'm working on the adrenaline, sir.
Okay.
Adrenaline hormone.
Yeah.
If we have, artificially into a body, induced our body, is it having any neurological effects?
Of course, yes. Artificial things always it has like a neurological effects actually. That, due to this neurotransmitters differences, definitely you'll have a lot of neurological defects actually.
And it causes any severe effects or minor effects, sir?
It's a minor effects actually, because severe effects, when it comes only, it affects the central neurons like a spinal cord or something, because neurons are like these are like just peripheral neuron, just it affects just locally actually. You'll not get like severe effects when you do something on this region, like pons or spinal cord and those regions, that's a problematic area. But other neurons, it's not, you'll not get like a severe effects actually.
Sir, are these affecting our vital systems, sir? Vital body system in regulating their physiological activities?
Yeah, for example, I mentioned, as I mentioned, amyloid lateral sclerosis. Actually, it's one of the genetic based actually, but otherwise, I can say in layman term, paralysis. It affects our regular body. For example, certain hand or certain part of body, it's paralyzing. Even sometimes face. Sometimes it also affects the locally actually, not like a central. Paralysis in whole, it affects the whole central. For example, stroke actually, it affects the whole nervous system. If anyone gets stroke actually, but in other cases, it affects locally some muscle paralysis, like due to some I can say is neuromuscular disorders actually. Some neuromuscular disorders are local, and still there are a lot of research going on to treat those locally affected neurons, neurological disorders. If I remember correctly, there are a lot of companies working on giving electrical impulses to rejuvenate the neurons, to activate the neurons actually. That's synapses in the neuromuscular regions. So I think it's locally as well as centrally. Sometimes it depends on the injury and part of injury, type of injury.
Thank you.
Hello?
Thank
Hello?
you, sir.
Yeah.
Hello?
Yeah, I can hear you.
Sir, is it Prabharan from Nanda College of Pharmacy?
Yeah.
Sir, one more question. You told laser confocal scanning microscopy.
Yeah.
Is it available in India?
Yeah, of course. A lot of
Could you please where is it?
As far as I know, all the IITs, they have such facilities.
Okay, sir.
Because confocal is not like a new microscopy.
Okay, sir.
But other microscopy I mentioned, like advanced microscopy, is like a new, but the 10 years old microscopies. Advanced microscopy is called STOME, stochastic optical microscopy. That resolution is 2 to 5 nanometers. But
Okay.
confocal resolution is 250 nanometer. It is available in many parts of India. Actually, I think each and every institute, they have a very good confocal. Then, to be frank, what we have it in European institutions, Indian institution has more advanced version of confocals actually. The problem is India, in the past 10 years, Indian IITs, Indian institutions has developed a lot, and in the past 10 years, they bought a lot of novel instruments, new instruments. So everything is like more updated than abroad actually. So a lot of institutions, all the IITs they have. You are in Nanda College of Pharmacy in Erode, right? So you can explore. IIT Madras is a bit far. Maybe you can explore in IIC Bangalore. Actually, they have a safe facilities also for this kind of central facilities. Probably in Salem, maybe Periyar University, they must have. Maybe they are not operating, but they must have this kind of instruments actually, confocal microscopes.
Okay, sir. Sir, one more question, sir.
Yeah.
Sir, your delivered brain transport of polymeric nanoparticle.
Yeah.
That is it possible to treatment of Alzheimer's disease?
Yeah, of course. That actually, the polymer itself is not like a therapeutics. Actually, the polymers is the polymers I shown using this polymeric nanoparticle. We used to encapsulate drugs, or we used to conjugate more specific materials like more specific antibodies or peptides. We conjugate with these polymers to cross actually. Initially, why we check the polymeric nanoparticles, whether the polymeric nanoparticle, what is the stability and how it behaves inside your brain. whether it's crossing, whether it has a good interaction, whether it has a good biocompatibility. That's the reason we did a preliminary experiments. But later, we encapsulate drug molecules. We put drug molecules to encapsulate them. We can treat Alzheimer's disease and things. But the problem is to treat Alzheimer's disease, the different mechanisms, the alpha, the beta fibril, the crystals present in the brain. Still a lot of research is going on actually to treat these Alzheimer's diseases more locally and using drugs. So still a lot of research actually is going on this particular area. Yeah.
Sir, one more question.
Yeah.
Sir, one more question.
Yeah, please.
Sir, any possible alternative model for your studies? Hello?
Yeah, you mean which alternative studies?
Sir, like now you are using rat model.
Yeah.
Any alternative models? Why you are using mice?
Exactly.
And also,
yeah, I'm sorry. There are a lot of alternative models. For example, of ethical issues, even in India, not only in India, many. I think I'm muted. Hello, can you hear me?
Yes,
yeah.
sir.
Yeah.
Yes, sir.
As I mentioned, especially for the neurological diseases, even for cancers, these days, there is a lot of models actually. Developing models itself is a recent trend nowadays. There are different kinds organoids models actually, like a patient-specific organoids. For example, we are exploring the cells from the animals actually, but these days, what many researchers are doing, even I'm doing actually those things. We submitted papers to Science Advances. The creating a models actually, like in vivo mimicking models using a patient samples actually. That's the recent trend is going on in the area of cancer, in the area of neurodegenerative diseases, any sort of areas actually, because taking directly the patient samples and prepare like a patient tissues, like it's kind of organoids, and using your materials to test actually, because using animals, always there is a interspecies variation. Sometimes in vivo is giving a very good answer, but when it goes to the phase 1, phase 2 clinical trials, it's getting a lot of failures actually. It's a big disadvantage for the drug discovery. That's the reason is patient derived, like xenograft models, patient derived organoids models are more alternatives for this kind of things I present just. And there is a lot of there's a lot of ethical issues, particularly in case of neurodegenerative diseases actually. As I mentioned, the papers we submitted, in that I used human cells, most of the human cells, but I couldn't use human microglia actually, because getting a human microglia, it's highly ethical issues. And actually I'm trying actually my level best to use human microglia. That's why I was talking with one of the big medical center in Pittsburgh, USA, University Pittsburgh Medical Center, and I think they agreed to work with the human samples. I think in the future we may access human samples, human microglia, or certain things. But in case of cancer, you can use human samples. It's very easy actually.
So good morning, sir. I have one question, sir.
Yeah.
Sir, this is a very nice presentation, sir, and very good informative also.
Thank you so much. Thank you.
My name is Mahendra, sir.
Mahendra,
Actually, nano materials and these nanoparticles the good in the therapeutic effect, but even as well as it's nanoparticles are toxic to the tissues also.
Yeah, that's why.
And this is it is oxidative stress or inflammatory markers or whatever it is, cytokine production. Then how do we decrease these problems in the nanoparticles? What are the methods or what are the alternative ways to overcome these problems? How?
Actually, that's regarding the nano safety. That's the reason I presented last 2 slides about nano safety. I'm saying nano safety. It's another way. It's called nanotoxicology. As you said, like ROS generations, cytokine generations, and everything is because of certain nanoparticles. Actually, that's the reason most of our research we are utilizing polymeric materials, which was approved by US FDA. It's shortly called as GRAS formulations, actually. So that's the main reason we are using a polymers, actually. If you want to reduce those toxic effects, you should not use, as a biologist I'm speaking, you should not use inorganic nanoparticles, actually. Inorganic nanoparticles and metal nanoparticles particularly. I think I have a quite interesting story to tell about silver. You know, silver, I think long back there is a guy from US, actually. Yes, I suppose in US, I don't know exactly which country. He got argyria. Argyria means it's like a blue skin. You know, this man, what he did, he continuously consumed silver ions for a month, actually. After a few months, he got like a argyria, like a blue skin and something like this, because when silver particles, it bounce on hemoglobin, and it he got such color. And I think afterwards he also died, actually. So you have to choose the materials, the like more biocompatible, biodegradable materials, actually. That's the reason we are more sticks with the polymers. Or these days, a lot of research is going on siRNA, RNA, something like this. You have to use the physiological relevant things, actually. Don't use metal or oxide nanoparticle like copper oxide, blah blah. These things, actually, don't use it, actually. And also another thing, you know, you ask me like how to prevent, actually. One thing, you have to choose the materials. You have to choose the polymeric material which is approved by USFDA or something else. And another case, still we are exploring, actually, how to prevent this the nanomaterial issues, like regulatory issues like aggregations, and so this is still we are exploring these aspects, actually. I hope I answered your question.
Oh, yes, sir. And which nanoparticle size is the best
Excuse me, sir.
therapeutic? Yes,
Please,
sir.
excuse me, Dr. Murali, sir.
Yeah.
Sir, there are so many people, sir. They are ready to ask the questions. So
Okay.
if you are answering the questions somewhat faster, it may be an good, sir.
Yeah.
Because so many people, sir, they are waiting.
Okay.
So I'm also requesting participants to ask your questions directly, and you can get your answers, please. So sorry, sir, because we are getting so many questions from YouTube's also, YouTube visitors also,
Yeah.
and till now some 3,000 people, sir, watched our YouTube live, sir. So it was
Okay.
very good achievements in our
That's great.
live seminars. So we are successfully that 3,000 people are watched, sir, till now.
Luckily, you mentioned this 3,000 numbers after my seminar presentation, because if you mentioned before, I maybe like get shocked, and so 3,000 is really huge, actually. I never presented in front of 3,000 people. My seminars. Okay. Yeah.
Okay. In our Zoom meetings also, at this 2 hour times also, at all the time that 300 people are there in the meeting, sir. And
Okay.
in YouTube's also that several people are watching, and still they are watching, and many people they are giving a good comments also, sir.
Thank you so
So
much.
my sincere request is that as many people they are trying to ask the questions from starting itself. So if you are going somewhat fast, means it may be on a good, sir.
Yeah. Sure. Definitely.
Sir, I have a question.
Yeah.
There are different sizes of nanoparticles. How do you separate the nanoparticles by size?
Actually, when you're that's a big challenge in a synthesis. Actually, when you're synthesizing, for instance, certain nanoparticles, like a metal nanoparticles, you know, you produce the poly. That's what we used to say is a polydispersibility index, PDI. Actually, in case of metal nanoparticle like silver nanoparticle, the dispersity index is wide. But in polymers, you know, you get like you cannot get more specific, like 100 nanometers exactly, but you get like plus or minus. That's why I mentioned in my table also plus or minus. Actually, you can able to synthesize 90 to 110. Actually, that's why we used to mention like 100 plus or minus 10. So most of the polymeric nanoparticle, you can define the sizes by different reactive conditions. But in metals, it's really difficult to make it actually. Metal means I'm not mention gold, because gold you can able to synthesize more highly monodisperse nanoparticles. But in silver, we had such a difficult, and iron nanoparticles actually, that also is a huge polydispersity. You cannot, that's a synthetics challenge actually. I can say I cannot like explain you specific methods, but it's a really challenging one actually. Synthesize.
Thank
Hello,
you,
good
sir.
afternoon,
Very
sir.
informative.
Hello.
Yeah.
Hello, good afternoon, sir. My name is Devi.
Yeah. Devi.
Sir, can we use nanoparticle treatments in the chromosomal disorders like Down syndrome, Edwards syndrome, and Patchu syndrome?
Actually, this is still in a research. I think you explore the PubMed. You know, you can see very tens or hundreds of papers, only tens of papers actually on this particular chromosomal disorders. Recently, very few researchers they started exploring this kind of chromosomal disorders using a nanotechnology. I don't know how far is clinically possible, but few research is going on at this moment actually. Even in PubMed or Web of Science, if you search articles, you cannot find many articles actually. Very few tens of articles you can able to find actually. So I think it's still in its infancy.
Okay, sir. Thank you.
Hello, sir.
Yeah.
Myself, Nino, and I just want to know that it will work on CNS disorder or not.
Which one?
No, nanoparticles, sir.
That's a good question actually. That's a big challenging. It works in CNS disorders or not. So far, there are some successful achievements. You know, as I mentioned, different mechanism, size-based mechanism. The lesser than 10 nanometer size, 50 nanometer
I did
size.
not hear.
Sorry?
Which one, sir? Sorry.
Yeah.
Which one mechanism you were saying that?
I mentioned in my presentation, one is like the receptor mediated mechanism actually. So that way, you know, you can conjugate specific receptors like brain specific receptor like transferrin. We also explored folic acid receptor specific delivery. So there is a possibilities actually, but apart from nanoparticles, you can consider like nanotechnology as a whole to treat a brain disorders. There is a lot of nanotechnological approaches to treat to brain diseases. Even I proposed for my startup company certain nanotechnology based actually, not like nanomaterials and nanoparticles. Using a nanotechnology, you can explore, you can target, you can treat, or you can circumvent this blood-brain barrier.
Thanks a lot, sir.
Yeah.
Anybody else?
Hello.
Hello. Yeah. Hi.
Sir, good morning, sir.
Yeah. Good morning.
Myself, Vinoda, speaking from Nizam College of Pharmacy, Hyderabad.
Okay.
And see, I have one question, sir. For some herbal extraction purpose, can we use the silver nanoparticles, sir?
I know a lot of papers are I saw in that way actually.
Okay.
Actually, why they are using I saw like a lot of papers they explains about natural silver nanoparticles or something. You know, further you have to use silver nitrate as a precursor, right? So what is nano? Because you're reducing using some kind of catalyst actually. So herbal materials has a lot of catalysts actually, alkaloids, you know, lot of catalysts actually present. So when you're introducing silver nitrate, somehow some materials will reduce and it forms silver nanoparticles. Actually, that's one of the conventional one, and I don't know how far it's effective in therapeutic actually. To be frank, I don't accept that way actually. To I don't even accept silver in a therapeutic, maybe antibacterial something, but still maybe localized application would be okay. But
What are the problems will raise in that project?
So which means that you're doing the project, and actually, I cannot say about the problems. Actually, it depends on your own interest, and depends on things. But
Out one question, sir.
yeah.
Hello, sir.
Yeah, I can hear you.
Sir,
I cannot say like it's a problems. Actually, you can get a nanoparticle, I'm sure about it, but those nanoparticles is not only silver nanoparticle. Maybe it bonds with different alkaloids which is present in the herbals, different terpenoids, many things actually. So the effect what you're getting, you cannot claim that it's only because of silver. It because of many things bound on the surface of silver nanoparticles. So that's not a problem actually, but I cannot say problems, but
okay, sir. Especially I'm choosing for that cancer purpose. So the silver nanoparticles are suitable for that herbal extracted drugs. So that's why I'm
that if silver herbal extracted silver nanoparticles, if they are treating cancer, means it's not because of silver, it's because of your herbal constituents. A lot of the materials are present on, and also nano. When it becomes nano, it becomes like a more active surfaces actually. So in the active surfaces, a lot of biomolecules and these macromolecules will bound. So if it is treating cancer, definitely it's not because of your silver. It's because of some herbal alkaloids or something else. This is my conclusion. I don't know. Maybe different people have different suggestions, but yeah, because silver is not like therapeutic one actually.
okay.
Yeah.
Thank you, sir. Thank you, sir.
Thank you, madam. Anybody
Hello,
else?
sir.
You have?
Hello, sir.
Yeah.
Hello, sir. This is Dr. Munija.
Hi, Dr. Munija.
Sir, actually, can you suggest any career opportunities after PhD, sir? Recently, I finished my PhD from Kakatiya University.
Okay. That's a more general question. As I suggested, what's your interest, like academic or industry, or do you want to go in other way, like consultant?
No, sir. Actually, I was interested in research, sir.
Okay, then you can if you're interested in research, like I feel like you want to do like a research or academic side something. So you can find a postdoc. I don't know area of research, but you can find a postdoc.
My area of research is GRDDS, sir, gastroretentive drug delivery system.
Okay, that's a very nice approach actually.
Yeah.
So you can find a postdoc and you can explore this way, or maybe I would suggest to you focus. Don't go like exactly same like your PhD, but slightly deviate your research area in your postdocs. Then you explore different areas so that you get a lot of ideas at the end of your PhD. And this is my suggestion actually. So you can find a postdocs. I think the sources I mentioned, your Axis, Nature Jobs, and you also contact professors from universities. Even you try India also providing a lot of fellowships from NPDF, National Postdoctoral Fellowships, and OPDF, Overseas Postdoctoral Fellowship, USPDF. So you can. If you bring your own money, everyone will accept actually. So you can explore it actually. And I don't know how many publications, because nowadays it's completely saturated, not like 5 years, 10 years back. So you need you need publications to prove yourself.
I'm having 15 publications, sir, up to now.
Oh, that's good actually. I always I'm sorry to say this. I'm not believing the numbers actually. I believe the quality of the publications.
Yeah, all are SCI indexed, sir.
SCI, even again, I'm maybe what is the impact factor of publications and something like this, because I remember 10 years back we used to struggle to publish in European Journal of Chemistry. I think Dr. Alagarsamy sir, he knows very well. But nowadays people are publishing in a JACS, Angio, Angenwanti Chemi, just like that actually. So because these days people are publishing like anything, so you must have a unique CV to be accepted.
Okay, sir.
Yeah.
Hello, sir.
Yeah.
You said that there are industrial PDFs also, no, sir?
Yeah.
Can I know about that, sir?
You can explore the websites of AstraZeneca, Novartis. There's some big companies actually. So you can explore those companies. They more often they used to advertise industrial postdocs. So that way you have to explore the even Bayer. I worked sometime in Bayer. Actually, Bayer is situated in everywhere in the world. So Bayer also offering some postdocs. So you can choose, you can select the website. Otherwise, our Sundar Pichai will help you. You can search through Google, and you can you can put the keyword and you can search option.
Okay, sir. Thank you, sir. You are very patient and answering all our questions. Thank you very much.
Thank you so much. Thank you.
Thank you, madam. So anybody else? Do you have any questions? So now I am requesting our principal sir, Dr. V. Alagarsamy sir, to conclude these sessions. Principal sir.
Yes, thank you, Dr. Saravanan. I think you have done your job. Dr. Murali have done the job excellently. All the participants are very happy to attend this session. And about 3,000 views were there in the YouTube. I think it is one of the great presentation that he has made in this big occasion. We also felt very happy to see these participations, because when we started this topic, we felt that many participants will be there because this is of the research topic. It's not a common topic. So we thought, but still we wanted to go presentations in a different topics, so that nowadays the students simply wanted to complete B pharmacy. So after B pharmacy wanted to go for GPAD. Area they are concising. So they are not going in a different manner or exploring the unexplored things. So if we have such a lectures, we thought it may open their eyes. So with that view only we have approached you, and really you have accepted our invitation without any hesitation. And all the participants felt very happy with your patient's presentation, and all the questions were answered, because you are one of the friend for us. We have also become friend to all the 3,000 participants. So hope in the coming days, Dr. Murali will associate with us, and we'll have many more sessions like this in the coming days. Thank you very much, Dr. Murali.
Thank you.
And
Thank you so
sparing
much.
for all the time. I also thank all the participants and management for giving us this opportunity. So now I request any one or two participants from other college other than MNR College of Pharmacy can say a few words, or one or two participants I request about this session. You can say a few words.
Sir, good afternoon, sir.
Sir, good afternoon, sir.
Yeah.
One by one.
I am Gogul from JK Nataraja College of Pharmacy, Namakkal, Tamil Nadu, sir.
Tell me, sir.
Sir, good presentation, sir. Murali Kumarasamy, sir. I would like to thank very good, valuable information shared at this time of pandemic situation, sir. And I would like to thank MNR College of Pharmacy, sir.
I think just for the information, Gogul, I'm going to present in your college also. Dr. Kapoor sir, he invited me to give a talk, so probably within 2 weeks' time, I'm going to present in your college also.
Okay, sir. Okay, sir. I am waiting, sir, for your kind of information and your speech, sir.
Thank you so much.
Gokul, you are in which course?
Gogul?
Sir?
You are studying which course?
Sir, B Pharm, sir.
Okay, very good. Really, our aim is satisfied by seeing the participants like this, because the B Pharmacy students come forward to attend this type of sessions. So that is our main motto to organize. So really very felt happy. Any more from different other state? Anyone from
Okay, sir. Thank you, sir.
I think
This information shared by your professor, Dr. V. Alagarsamy, sir. I would like to thank, sir.
Okay, thank you, sir.
Hello.
Good afternoon, sir. I am Navina from Balaji Institute of Pharmaceutical Sciences, Warangal. Dr. Murali, sir, you gave such a good information and informative, fruitful session, sir. Thank you for replying every question. Thank you for your patience, sir.
Thank you so much. Success requires patience, so that's I believe a lot actually. So
thank you, sir.
Good
Sir.
afternoon, sir. This is
Sir, one
Dr.
request
Satish
from
Sharankar.
my side, sir.
Yes.
Sir, kindly share this type of webinars and seminars to our following registered mail participants, sir. Kindly share.
Definitely, Google. We will send that next webinar details also to your email ID, whoever is registered for these seminars. And
Okay, sir.
our management as well as our principals are very enthusiastic, and they are encouraging us to do more and more webinars. So in the next coming week also, we are planned for such 2 webinar from the eminent personalities from the various part of the world, that in the different countries. As we are conducting on an international webinar series, we are planning for things. So definitely we will send the invitation to your registered email ID. So there is no doubt. We will send you.
Okay, sir. Thank you, sir.
I think with this feedback, we can close the session. So thank you very much. As Dr. Murali told, patience is the first thing required for the success. I do remember very well about almost 10 years back, Dr. Murali was searching for the higher positions after M-Pharmacy. But that time he was very clear. He wanted to do PhD in abroad, and he wants to do PhD in a something new area, not like M-Pharm chemistry he has completed. He want to go do the M.Pharm pharmaceutical PhD pharmaceutical chemistry. He wanted to do PhD in some different area that is unexplored, so that he can be entered in the new era. So now I am seeing him after 10 years. He could achieve what he wants. So the patience, listening, and the constant efforts only make any man to become reach any heights. So that is understood from Dr. Murali. So really we felt proud of you, Dr. Murali, for having such association. And you are one of the inspiration to the all participants. So definitely they will take your words and they will come forward to enter into the new era of the science and make our pharmacists proud in the scientific community. So
Thank you
with
so
this,
much,
thank
sir.
a few words. I thank Dr. Murali for sparing all the time and participants also. So in this occasion, for his valuable time spending, token of appreciation, we would like to present a certificate for his presentation. I request Dr. Saravanan to share the certificate.
Yes, sir. I already shared in the screen, sir. I think hope you can able to see.
No, it's
Yes, sir.
not yet, sir.
Not at
Not yet.
Sir, just a minute, sir. We are working on.
Sir, one more request, sir.
Sir, I think we shared, sir. I think in YouTube also it is coming, sir.
Oh, okay.
Sir, Murali, sir, can you able to see?
I couldn't see actually.
You couldn't, sir. Can you stop your screen sharing, sir?
Yeah, I can see. Yeah, thank you.
Yes,
Thank
now
you.
it has come, the certificate. Some participant is asking doubt. I think you can display Dr. Murali's email, sir, so that let them be in touch with further communication.
Definitely, sir.
Dr. Murali, sir, kindly accept this certificate as a token of appreciation from our institution. Kindly accept it, and thank you very much, sir.
Thank you so much, sir. Thank you so much for a kind invitation. I'd like to extend some more things, some more words actually.